Okay, what if I told you that pretty much everything alive is built by the same tiny, microscopic team? Yeah, I'm talking from the simplest little bacteria all the way up to us. It turns out a group of just 20 molecules are the master architects behind it all. These are amino acids, and they are, quite literally, the foundation of the entire biological world. So here's a riddle for you. What do your own muscles, super strong spider silk, and some of our most life-saving medicines all have in common? I mean, think about it. You've got the raw power of muscle, the insane tensile strength of spider silk, and then the delicate complex chemistry of medicine. What on earth could possibly tie all of those together? Well, the answer is proteins. And every single one of them, I don't care how simple or crazy complex it is built from a set of just 20 core building blocks. And those are our amino acids. It's kind of mind-blowing, right? This small handful of molecules is responsible for the staggering, almost unbelievable diversity of life we see all around us. So that begs the question, right? How can just 20 molecules create, well, everything, all this incredible variety? The secret, it turns out, is in their fundamental design. So let's dive in and break down the blueprint of these essential building blocks. Okay, so take a look at this. Every single amino acid shares the same basic structure. A really good way to think about it is like a toolkit, you know? Every tool has the exact same handle, which makes it super easy to grab and link them all together. But each one has a totally different head, a different functional part that does a unique job. All right, let's break down that toolkit. It all starts right in the middle with a central carbon atom. That's our anchor. Now attached to it, you've got three standard parts. First up, you have the amino group. Then you have the acid group. Put them together and, surprise, you get the name amino acid. There's also a simple hydrogen atom, but it's that fourth attachment, the R group, where all the magic really happens. Seriously, this R group, it's everything. This is the one part that changes, the variable bit that gives each amino acid its own unique identity. I mean, it could be something as simple as a single hydrogen atom, or it could be this huge, complex structure. This side chain basically defines the amino acid's personality. Is it going to be acidic? Basic? Does it love water or hate it? All of that is decided right here. This is the source of all that diversity we were talking about. Okay, but it gets even cooler. Because that central carbon has 4 different things attached to it, amino acids have this really fascinating property called chirality. And the easiest way to think about that is just handedness. Just look at your own hands for a second. They're perfect mirror images of each other, right? But try to lay one directly on top of the other, palm to palm, fingers aligned. You can't. They're not superimposable. Well, it's the exact same idea with these molecules. You have L isomers and D isomers. They're mirror images for sure, but they are fundamentally different shapes. And this is where we run into one of the deepest, weirdest mysteries in all of biology. For reasons we honestly still don't fully get, life just chose a side. The proteins in your body, in every plant, every animal, every living thing on this planet are built almost exclusively from the left-handed L amino acids. It's this universal signature of life as we know it. You can think of it like a cosmic coin flip that landed on left billions of years ago, and it's been that way ever since. But wait, there's more. Their shape isn't the only trick these molecules have. Amino acids are also kind of like chemical chameleons. They can actually change their electrical charge depending on the environment they're in. And this whole ability to change charge, it's all about pH. So, if you stick an amino acid in a really acidic solution, it'll grab an extra proton and become positively charged. If you put it in a basic or alkaline solution, it'll lose a proton and become negatively charged. But right there in the middle, at a more neutral pH, something really interesting happens. It becomes something called a Zwitterion. Now that's a cool word, right? It actually comes from the German word for hybrid, and that's because it has both a positive charge on one end and a negative charge on the other at the same time. So those charges cancel each other out, giving the whole molecule a net charge of 0. And this is the form that amino acids are usually in inside our bodies. So, if the charge changes depending on the pH, well, that must mean there's one very specific pH, a precise sweet spot, where that perfect neutral Zwitterion form exists. And this curve right here, it tells the whole story. It shows you the molecule's net charge as we change the pH. So look, way over on the left, at a low pH, the charge is positive. Way over on the right, at a high pH, the charge is negative. But do you see that exact spot right in the middle, where the curve crosses the 0 line? That is the sweet spot. We call it the isoelectric point, or PI for short.
So the pI is simply the exact pH where our amino acid has no net charge. It's perfectly neutral, and this neutrality has a really cool consequence. At this specific point, the molecule is least soluble in water. Now, that might sound like a random chemistry fact, but it's not. It's an incredibly powerful tool that scientists use all the time to separate and purify proteins.
And guess what? This all comes right back to that all-important R group. Check this out. An amino acid with an acidic side chain, like aspartic acid, has a really low isoelectric point, down around 3. But then you take one with a basic side chain, like lysine, and its pI is way up there, almost at 10. It just goes to show you that R group completely dictates how the molecule behaves electrically.
Okay, so why should you care about any of this? Why does the pI matter? Because this is a fundamental concept with huge, real-world implications. It's absolutely crucial for scientists who need to separate proteins in the lab. It's a major factor in determining how proteins fold up into their complex 3D shapes, which decides their function. And yeah, it's even a key principle in designing new medicines. It's a simple idea with incredibly powerful applications.
So, let's recap. We've seen the basic blueprint, we've talked about that mysterious handedness, and we've seen their chameleon-like ability to change charge. We basically understand the individual bricks now. But how do these bricks actually link together to build the incredible, complex machinery of life? Well, that's the next part of the story.